Welcome to the second episode of Better Together. Today we're joined by Thomas Gundersen of Vitas, and they're the developers of the dried blood spot test for COVID-19. As usual, you can ask a question. There's an Ask a Question tab below, so just put in all the questions that you have, and we'll try to go through them during the end. And there's also a comment section, but don't confuse those two and just ask a question below. All right, so we can just hop right into things. Welcome. Thank you for being here with us.
Thank you.
Yeah, so first question, could you just introduce yourself and Vitas and what you do?
Yeah, so my name is Thomas Gundersen. I'm CEO of Vitas. So Vitas is a CRO. It's a contract lab doing chemical analysis. And we serve pharmaceutical companies, biotech companies, primary healthcare, and a lot of startups around the world. So our company, Vitas, was established in 1994. So this is actually 26 years ago. And it was during my my master, where I had an assignment from a couple of professors at the Institute of Nutrition Research. And the task was to develop an assay for vitamin A in biological samples. So I did this, and I did it pretty well, I think, because like 6 months into this master, they gave me a large sum of money, like more than 1 million Norwegian kroners, to establish an automated system for analysis of fat-soluble vitamins in blood. And I think I also did that quite well, because the same year we signed a contract with Fürst Medical Laboratory in Oslo, and to do fat-soluble vitamins for samples coming from the primary healthcare in Norway. And actually, at this time, there was not possible to do this for doctors in Norway. So we kind of established the market and made this methodology available to the Norwegian population. So this was including vitamin D. And as you know, and people know, that vitamin D is a really big market today. And it's kind of cool that we actually were the first one in Norway to ever do this, and it has grown to really large dimensions. So over the last 25 years then, we have developed more than 1,500 methods. And we have grown to 22 employees, and we have like 35, 36 machines, robotic systems, doing analysis 24/7. And currently we receive close to 20,000 samples from all over the world every month.
So these are 20,000 samples within the dried blood spot testing?
Altogether. Most of them are dried blood spots.
Yeah. So exciting. And actually, could you tell us a little bit more about dried blood spot sampling and testing? What is that, and how did it come about?
Yeah, so when you go to the doctor, you usually stick a big needle into your arm and draw like some 4, 5, 10 milliliters of blood into tubes. And often there are many tubes, So on the contrary, dried blood spots is just one single drop collected from the fingertip and dripped onto a piece of paper, special paper, and dried, and you can send it by regular post to our lab for analysis.
And it performs the same tasks as when you go to a doctor and get your blood.
Most of the assays you do at the doctor is also possible on dry blood spots, but there are some things that's not possible.
Yeah. And how did this methodology really kind of fit into the COVID-19 situation and the testing that you've developed, essentially?
Well, I think especially for COVID-19, where people were told to stay home, you cannot travel, go to the doctor. There were quarantines, and people were also afraid to infect each other. I think this kind of platform is very well suited, because this is actually the only offering where you can stay at home, get a kit to sample at home, send it in post, and get the analysis online. Yeah.
And what kind of inspired you to use this method? So apart from the fact that it was more easily accessible in that sense that it could be distributed to a lot of people, were there any other kind of inspirations that brought about?
Well, I mean, there was a crisis. Businesses were shut down, I was very worried that my company would go shut down, that the income will stop. So I figured out what can I do about COVID-19, because there were a lot of funds available from the research council and so on.
Mhm.
So I wrote an application together with one of the professors, and we supplied it in Thursday, the 26th of March. And in the morning, the day after, in less than 10 hours, we were funded. So
Wow.
I think for me, this is like a world record. So I think it was really fascinating. And since March, then we have been working really hard to develop a fast-track method for COVID-19 serology
Mm.
in dry blood spots. And now it's finished, and we are launching this in the market.
Yeah. Could you just talk us through about how does this kind of actually work? How do you test for antibodies in this sense?
So we have all read about this testing in the
Mm.
newspapers and seen on TV and so on. And when you get infected by a virus, like the SARS-CoV-2 virus, causing the COVID-19 disease, you will, from day zero after infection, you will start to have growth of viruses in your upper respiratory tract, right? So then you can take a sample from the nose or the mouth and do virus analysis. Well, then will disappear after 10 to 14 days, the virus, and then the body will start to produce antibodies to kind of strengthen your immune system against reinfection of the same virus. And these are called immunoglobulins. And there are two different types, IgM and IgG. So IgM will start to appear in the body after 7 days and disappear after 14 days.
Okay.
And then after 14 days, you will have production of IgG. And this will last for probably 1 to 2 years. And this is your immune defense against reinfection of the SARS-CoV-2 virus.
Okay.
And this is what we measure on the dry blood spots.
Mm.
So if you test positive for IgG, then we are sure that you have had the COVID-19 disease previously. At least 14 days, maybe 20 days since you were infected.
Mm-hmm.
Yeah. And it's very likely that if you have high levels of IgG in your blood, you will be protected to reinfection totally. Or if you get infected, which you can, you will be much less sick, right? So this is the kind of the important thing about this test.
Interesting. And there is also something that you mentioned about the sensitivity and specificity.
Yes.
Could you just tell us a little bit about that?
So these are two really important terms when it comes to this type of testing, serology. And so the sensitivity is the true positive rate. So imagine you have 100 people that you are really sure had the COVID-19 disease, and you apply samples from these 100 people in our assay. And if we then find that 96 of these 100 have had the disease, the sensitivity is 96%, right? So we kind of miss 4. So the true negative rate is that if you have 100 people that you are quite sure never had the disease, maybe if the samples are collected like 2 years ago, prior to the pandemic, and we find that 4 of these had the disease, which is not true, the specificity is 96%.
Yeah.
So our assay has a sensitivity of more than 96% and a specificity of more than 98%, which is really good.
And actually just kind of going more into kind of how you collected, so were the samples for people, like the negative samples that were collected before?
Yeah. So we have negative samples in freezer.
Yeah.
Because this is what we do. So we had a lot of samples. So we collected all samples for negatives. And for positives, we did recruitment through social media. So we used our social media network. We kind of asked people to join us in what we like to call the crowd science approach, you know. And people were really eager to travel to Forskningsparken and show the evidence, the certificate or the journal, the medical journal, to prove that they had the virus identification. And then they gave venous blood and dry blood spot blood. And we then compared these two to validate our assay.
Okay. And how many samples, because I mean this is kind of something that's ongoing.
This is ongoing.
What are kind of your hopes for this at the moment?
So we are aiming at least 100 people, positive people.
Okay.
So we are now at about 72. So it's going well.
Yeah.
So people are joining all the time, and soon we will be at 100 people, positive people.
Yeah. And I really like the term crowd science. Could
Yeah.
you let's dive a little bit into that? I
Yeah,
think
I mean, I
it's
think all people, we have all heard of crowdfunding, right? So this is a way to have microfunding, like 1 million people are contributing with 5 kroners each, then you can fund like the development of a new device somehow. And in the same style, I think we will see in the future what I refer to as crowd science, that people are contributing, like in this case with blood, to solve a scientific problem. So other kind of topics could be sleep disorders, for instance. So if you ask, so we have a large customer base, maybe 200,000 people. So we can ask them on social media, do you have a sleeping disorder? Yes. So would you like to contribute to this research? Yes. And can we use your sample, for this research? Yes. And then we can do really quickly studies on 10,000 people in 1 month, and then we can publish this. And I think this is a genius way to do crowd science in the future.
yeah, that's really cool. What inspired you to bring this out to the market? I think you kind of like
Yeah,
touched a
to save
little
my
bit.
company, one
Yeah.
thing, of course, to contribute to solve the problem.
Yeah.
We all need to contribute. If we will be able to conquer the disease and to get society back on the feet, I think. Yeah. So
Yeah.
that's those are the two main reasons. And I also experienced that the people working for me in my company really like this now, that we have a really nice buzz now in the company.
Yeah.
Everybody is really working hard to contribute to this problem.
Yeah. That's I mean, you guys must have been working overtime. And I mean,
Yeah.
even with our previous guests, we kind of asked a little bit about how you're coping, because is there like a strong demand? Are you finding any challenges within your business to cope with this? Or
I mean, for us, it has been, we did have actually an all-time high in March. Then we had a dip in April. Now we're back in business. And I think from this COVID-19 offering that we will do next week, I actually think we will see a massive growth in revenue in the company.
yeah,
So it's a good time for us, actually.
that's good to
Yeah.
hear. And I think there's also kind of something that I've been picking up a lot on is I don't know, I would call it I don't know if it's a misunderstanding or a controversy in regards to the word using it as
Home
test.
testing. Yeah.
Yeah. Could you tell us a little bit
Yes, I
about
think
kind of
there's
what's happening in that area?
absolutely. So there's a lot of misunderstanding. So when people refer to home testing, it's usually using like a pregnancy test device, like a plastic device. You prick your finger, you drip your blood, and then you will see this line appearing on this device. So in this case, this is a real home test. Everything is happening in the home by the user itself, application of the blood, the analysis, and the interpretation of the results. So this is not within the rules in Europe. So this was stopped. So our test is referred to as a home test, but it's actually not. It's a home collection device. So we send a kit home to the user. They prick their finger, they apply a blood drop to the paper, and then they send it back to the lab for analysis. So the important things, the analysis, the interpretation is done in a professional lab. And if you do it like this, you have a CE-IVD collection kit and a CE-IVD analysis in a professional lab, then it's within the rules and it's okay.
Could you just, what is CEIVD?
So CE is the kind of European certification
Okay.
mark. You can have it on electricity. I mean, everything sold in the European Union is must have a CE mark. And the IVD is in vitro diagnostics, which is referring to the blood analysis part of it. Yeah? So what we do is within these rules, and it's totally allowed to sell this service in Norway and in the rest of Europe.
Are these tests available, or collection kits? Are they available right
Yeah.
now, or?
As you know, we've been selling this prototype kit
Yeah.
in Oslo Science Park for a couple of weeks.
Yeah.
And it was really funny because like when we opened up, we sold 40 kits in 10 minutes, and in the same day we sold 250 more. So it seems like there is a market. And next week we will be launching the commercial product, looking slightly different from the prototype. I think you already posted this online, and it will be sold on bodymarkets.com, which is a partner that we collaborate with. So this will be the first selling point, but then there will be additional later on.
Yeah. And actually, people just ask questions if you have any. Just don't be shy.
And I would like to add, if there are companies like larger companies, like 20, 30, 40, 50, 1,000 employees, and the CEO or the human resource management would like to screen the employees, they can send an email directly to me, and we can ship like a big box of kits to this company.
Yeah. And kind of touching on kind of like this screening, are you hoping that it will kind of bring a little bit more, I don't know if security is the right word in society, or kind of what kind of effect are you hoping for within the general populace?
Yeah, I think a lot of people, I think today we could read in the newspapers that about 30, 40,000 Norwegian probably are infected, which is not a large number, but I think most people think am I one of those 30, 40,000 people, And I think people would like to know. And the only way to know is to measure, right? So I think it's important that there is an offering, not only to hospitals and doctors and so on, but to the general population. I think this will be the first offering of this kind that can really reach the entire country. Yeah. Yes.
And how long do you hope to continue with this? Because in essence, it is research, because you are trying to.
Yes. So the infection rate, so I think today, Folkehelseinstituttet, think it's 0.7% of the population that's infected. So the numbers that we have so far, 0.24% actually. But of course, this is not really comparable, because I think the people that have bought our kit is really motivated, and it probably means that they have been sick, right? So the chance of they having the disease is higher than in the general population. And also we have provided kits to persons that we know have been infected, but not only to them. These are not within the statistics, but to the family surrounding them, right? So mother, father, children, and so on. And of course, since there was one infected person in the family, it's more likely that some of the others also have been infected. So I think the number we have, 4%, is much higher than 0.7%, because it's more likely that we have infected in our cohort, in the group we have studied.
Yeah. I think that's really fascinating. Actually, for the people that you were testing and looking for these results, so you say that you gave them a test, and then also their family. Were there any, if you can talk about it, any kind of interesting findings that you found in there?
I cannot say so much.
No,
but
that's absolutely
I have
fine.
multiple mails from fathers, mothers, and they have been isolated for 8 weeks. They don't want to go out because one of them had the infection, and they are afraid that the rest of the family also was infected. But there is no possibility of measurements, right? Because you're kind of not in the risk group, then you are not offered analysis. But so they are so eager to kind of clarify if the whole family had the disease or not, because if they had, they can go out and break the quarantine, right? Go back to normal life. So I really think that this service, this offering, is really important. I think people will appreciate and do the analysis.
Yeah, I mean, I think it is a nice way, because I think also a lot of what's going on, I think a lot of people are anxious, a lot of people are a little bit nervous. So I do think that it is kind of a really wonderful initiative in, I'm not quite sure what the word is, how to put it into words. It's a wonderful initiative in including people in trying to make a difference, or learning a little bit more about what's happening and what we can do. So I think it's...
And I actually think when we have the vaccine, this service will be even more relevant. Because,
how so?
Because when you have the vaccine, you kind of actually forcing an infection, right? You inject a dead virus into your blood, And what happens then, or supposed to happen, is that you start to produce antibodies. You boost your immune defense, right? But we actually don't know if it happens, you know? So I think after the vaccine, people should actually do the test to make sure that they actually produce antibodies. And sometimes you maybe need 2 injections, right? And then, in addition, we never know actually how long the antibody levels last in your blood. So I think also it's relevant to do another analysis after 6 months, 12 months, and so on to see that you still are protected from reinfection of this virus.
Yeah. So, in relation to the test that you've given, 2 people have tested positive. Do you also do this where you kind of test them ongoingly, or
Yes.
how?
So all the people that tested positive, and then we have a certificate, confirmed positives. We hand out like 10, 20 DBS kits that they take home. And every week they take a new sample and send us the sample. So we have these timelines for multiple people, and we can see how long the antibody levels last in their blood. So this is part of the research.
Yeah.
And of course, very important for the individual to have to do the measurements on.
Since this is ongoing research, do you hope to publish it, or is there?
Yes, so we are applying to the ethical committee, REC, in Norway. So we will be allowed to publish this in scientific papers later on.
Yeah, cool. We have a few questions over here, which I can just take on. Here we go. So Astrid is asking, do you think the government should apply this test in a larger scale or on a larger scale?
Well, if it's only 0.7% of the population that's been infected so far, I think it's too early to do a massive population screening. But at some time point, I think it will be right to do this screening. And then I think our DBS platform has a really powerful distribution, and it's really powerful in distribution because you can easily send out like 50,000 letters, collecting samples at home, and get the samples back in the regular post system. In opposite to having like 50,000 people travel to the hospital to do a regular sample. So I think when staten, Folkehelsa, is ready to do this, I think they should approach us for such a service.
Yeah. And let's see here. Eleni is asking, when do you think we will have a vaccine, or what is your best guess?
I really wouldn't know, but I think I read in the paper today it might be like 21, 22.
So 2021, you mean?
So late 21, I think it's likely, but I'm not an expert on this.
Yeah. And actually, just something I kind of thought of when you were mentioning kind of like sending, since people are collecting blood at home, do you have any fears, like, you know, I don't know, something silly, like their dog might lick the test or something?
Yeah, I mean, it is a medical device, you know, so you need to respect this. Of course, you cannot contaminate the paper with something else. And if you do, you shouldn't send the sample. Then you should ask for another set. Right?
Yeah. Yeah, so I think for anybody trying this at home, just be careful. And let's see. So Elizabeth is asking, there have been some criticisms about how correct COVID-19 tests are. How correct is your test, I think?
Yeah, so this is kind of the sensitivity and specificity, you know. So the validation shows that it will correctly evaluate 96 to 98% of the people, right? So there is a small error margin, but it's really good. So in opposite, this home test, you know, this pregnancy type of test could have a sensitivity of 60%, right? Which is much. Then 40%, 40 out of 100 people will be wrongly evaluated, and that's not good.
No.
So 2 out of 100, I think it's good enough.
Yeah. And I think you're also mentioning that in a way it isn't a test. It is that you're collecting blood and then you're taking it back into your...
It is a test, but the test is done in the lab.
Got it. Okay.
So the collection is done home, and the test is like a professional assay test method done in a professional lab. And I would say that the important way of using this result is that if you are tested positive, then you had the disease. But if you're tested negative, you should behave as you have been up to now, avoiding having the distance, cleaning your hands and everything, because you might actually be you just have been affected by the disease and it will not show up on the test, right? So it takes about 14 to 20 days before it shows up on the test.
Yeah. Okay. Let's see. And Sigurd is asking, what other things can you test for using this dried blood test? Are there any exciting examples?
Yeah, also in addition to the antibodies against SARS-CoV-2, one can test, like, if you want to go traveling, for instance, the dengue fever, the Zika virus, like things you need a vaccine for before you go traveling to exotic places. And in addition to nutrients like vitamin D, omega-3 fatty acids. Or if you have diabetes, we can test HbA1c, is long-term glucose in your blood. So there is a lot of uses. In total, we have about 200 different uses on this platform.
Wow. So it's just kind of a side question here. So your company kind of does all these?
Yes.
Wow.
Yes.
That's
So
interesting.
actually, we receive samples from almost 75 countries now. 48 out of 51 countries in Europe, South Africa, US, Canada, Australia, and so on.
So this is both testing nutritional and kind of diseases.
Yes. So this COVID-19 DBS approach is not something new. Well, the COVID-19 is new, but the DBS platform is something we've been running for 15 years.
Yeah.
And as I told you, we receive 20,000 samples each month.
Yeah. So in that way, you have the experience within the methodology.
Yeah. So it's well established and very robust, and this is something we know very well. Yeah.
Yeah. Did you encounter any problems or challenges while you were creating this test? So even though you have done this process before and you've engaged in this, were there any kind of difficulties or nuances that you encountered coming to develop something to test for the antibodies and COVID-19?
So we have similar experience from other similar assays. So it was not that hard. The hard part here was to do it really fast. So we did it in 2 months, the development, which is really fast. And now we also raised a commercial company in 1 month. And it's high volume production, we have like these kits produced in really high volumes. And we have a system, infrastructure and a logistics system that will be able to sell like 100,000 kits per month. So it's kind of a fast track development of both the assay, but also the business coming up now next week.
Yeah. Fascinating. Let's see. So Vlad is asking, can the test be used for things such as STDs?
Sexual transmitted
Transmitted
diseases.
diseases.
Yes, the answer is yes.
Yes. Okay. Won't dive too much into that, but that can be a topic for another day.
Absolutely.
And let's see. Eleni is asking, are there other companies that have developed antibody tests, and what is the difference between your test and these other tests?
Yeah, there are like Fisht, for instance, our previous customer has a service for the primary healthcare in Norway. I think it's very identical to what we do. The only difference is the sample type. So when you go to the doctor or Fisht, you need to go physically and have this big needle into your arm. And so after that, it's the same thing. And in the US, there is a company called LabCorp, which is one of the largest clinical companies in the US, and they offer also from dry blood spots I've seen. So they call it a kind of a back to work service.
Okay.
So this is also what we think this might be used to, kind of a fit to work, fit to go to the gym, fit to fly, fit to travel, fit to hug, and so on, So I think people really need to know if they can go back to work, going back to hugging each other and so on. And this is something that this service can be used for.
Does it make you a little bit nervous, perhaps, in how people can translate? Because, can translate the results? Or do you have like a way of kind of streamlining what a result means at the moment?
So we have an online report, web report. So when you buy the kit, there is ID code. So this is an anonymous service. So the one that open up the kit is the only one that knows the identity, right? This is a number. So when you have the results ready, you log into this web page, you enter the code, and then you have the report. And it will say green, positive, or red, negative. So we do all the interpretation. So either you had the disease or you didn't have the disease. Yeah.
And let's see. Yeah, it's 2 questions left over here. And how do you know that it is the new virus and not the earlier? I'm not quite sure what that means.
I mean, there are some, this is, this.
There's 2 questions. One question, read the other question.
Oh,
Okay.
sorry. Thank you. There we go. Now I can understand this. What's the difference with this coronavirus and those that have been earlier? And how do you know that this is a new virus and not the previous virus? So I'm guessing it's within strains, perhaps, or?
Yeah, so this is the 7th coronavirus.
Okay.
So there's been 6 previous coronaviruses. So when we say something about immunity, it's based on the knowledge from the 6 previous coronaviruses. Then it's likely that you have immunity if you have IgG in your blood. So there are some small differences, but it's called the antigen. It's kind of a protein on the surface of the virus that is the basis for the assay. This is very technical, but this antigen is very specific. So it will only be present on the SARS-CoV-2 virus, So the previous one was SARS-CoV-1. That was the original SARS. Now we have CoV-2, which is a new subtype of the same virus. But the assay is really specific. There is a small chance for cross-reactivity to some of the previous ones, but it seems like, the sensitivity, the validation is 98, 96%. So this is kind of the accuracy of the assay.
Yeah. I'm guessing this was something that you also accounted for when developing.
Absolutely. But there is a small chance of cross-reactivity to all the viruses.
What does cross-reactivity
I mean,
mean?
that the assay will also give a positive if you have infection from one of the 6 previous coronaviruses. Yeah. But this is not very likely because this has kind of died out. So now we have the COVID-19 and not the older ones. Yeah.
Okay. And I'm just going to ask a little bit about the ethical component of this. How do you deal with that? So when you collect these, what happens to the samples afterwards?
Oh yeah. So first of all the people that contribute to the research project, they have signed an agreement that they consent, and there is an information letter, and they have signed. So they agree to do what we do in this project. Those buying kind of consent by buying, right? So after when we have done the analysis, we keep the sample for 14 days, and then we kind of burn it, disintegrate it through a third-party service that we use for all the biosamples we receive. Yeah. And as you know, the analysis is anonymous. It's only the person that contributes to the sample that knows the identity of this blood sample.
Yeah.
Yeah.
Nice. I think we are going to be rounding up. We're at the end. Thank you so much for amazing questions, and thank you for joining us. I think I saw a post on Facebook that these kits are still available, at least in Forskningsparken.
They will be available in Forskningsparken for a week or 2. Next week, they will be available at bodymarkers.com. And if there are some larger companies that would like to do a large package, of hundreds or thousands, they can contact me directly.
Perfect. Yeah. So thank you guys for joining us, and have a nice lunch. Bye.
Bye.